Hello everybody and welcome to our second lecture in Topics in Biology. In today's lecture we are going to be talking about the structure of protein molecules. I hope you enjoy the lecture. Proteins are very important and very instrumental in everything that an organism does at a cellular level. They play roles in many they have functions that include structural support, storage, transport of other substances. They play a role in the intracellular signaling between cells as well as movement, and they also play a role in defense in the case of antibodies, and they also act as enzymes in the cell to catalyze different reactions. Humans and all organisms have tens of thousands of proteins, and each one of them has their own unique function. Now, the function of the protein is derived from its structure, and proteins are the most complex macromolecules known because they have a 3D shape that we call the protein's conformation, and you can see that on the figure on the right hand side. All proteins are made of amino acids, and there are 20 amino acids that make up all kinds of proteins in a cell. We call them the monomers. Now, the polymers of proteins, or the polymers of the amino acids, are called polypeptides, and proteins can consist of one or more different polypeptides, as we will see in today's lecture. Now, before we go into the structure of the polypeptide, we have to understand the structure and the chemical structure of the amino acids. Now, all amino acids have four components that are attached to a central carbon that we call the alpha carbon. The alpha carbon has four groups attached to it. It has a hydrogen, it has a carboxyl group, and it has an amino group, and then it has an R chain or R group. And this R group is different between the different amino acids. And like I said, we have 20 different amino acids, and therefore we will have 20 different R chains. Now, depending on the type of R group, we can divide the amino acids into different groups. They have different physical properties. They also have different chemical properties that give rise to each characteristic of a particular amino acid. Now, depending on the R group, we can divide the amino acids into nonpolar amino acids. We call them the hydrophobic amino acids. We have the polar amino acids, and we call them the hydrophilic amino acids. And then we have the electrically charged amino acid or ionized amino acids. Now, going into a little bit of details, the nonpolar amino acids, they have a hydrophobic R group. Now, what does hydrophobic mean? Hydrophobic means that they fear water. They do not like water. And if you look here, here are some examples of these nonpolar amino acids. For example, the simplest amino acid known is glycine. And as you see from the figure, it has a very simple R group that is simply made up of a hydrogen. We then have alanine that has CH3 R group, and as you see, valine, leucine, isoleucine, and the different kinds of nonpolar amino acids. Next, we have the polar amino acids, and these have hydrophilic R groups. Hydrophilic meaning they like water, they like to interact with water. An example of serine, threonine, and cytosine, as well as some aromatic like the tryptophan. Third, we have the electrically charged amino acids, and these have functional groups that are charged or ionized. Basically, they have a positive charge or a negative charge at cellular pH. Now, cellular pH is in the range of 6 to 8 pH. Now, some of them are bases, and some of them are acids. And as you all know, a base is a molecule that accepts an H plus, a proton, and an acid is a molecule that gives off a proton. And as you see on the figure, we have 5 kinds of these electrically charged amino acids. 2 of them are acidic, and 3 of them are basic amino acids. Now, how are amino acids joined together to give rise to a peptide bond? Like nucleic acids that we spoke about in the previous lecture, the two amino acids come together and they join together via a condensation or dehydration reaction. And as you see from the figure on the right hand side, what happens is an OH group from a carboxyl side of the C terminus of the amino acid interacts with the H of the amino terminal of the N terminal of another amino acid, and a water molecule is removed. The resulting bond is called a peptide bond. Now, a polypeptide, basically, if you repeat this process of the dehydration or condensation reaction, you will end up with many amino acids being joined together by these peptide bonds, as you can see from that figure. And we call this a polypeptide chain. And attached to the backbone, there are various R groups. Now, polypeptides range in size from a few monomers or a few amino acids to thousands of amino acids, depending on the protein or the peptide that is being synthesized or being made. Now, like I mentioned earlier, a protein's function depends on its specific conformation. Now, a functional protein consists of one or more polypeptide that is twisted and folded and coiled into a unique shape, and we call this the 3D conformation. Now, protein-specific conformation determines its function. So here are some examples of protein functions. All right, basically, a lot of proteins like antibodies and enzymes, their function is to recognize and bind to a specific molecules. And if you look at the figure, the top figure, you can see antibodies binding to a foreign molecule that they recognize as maybe parasitic. And on the bottom there, you see an enzyme, and the function of an enzyme is basically to recognize and bind a specific substrate to facilitate a particular chemical reaction, all right, to catalyze a specific chemical reaction. That is why the protein's shape is very important in its function, and it plays a vital role in determining the protein's function. We have to understand various levels of the protein structure. Now, we have the primary structure, the secondary structure, and the tertiary structure. And all these are basically within a single polypeptide molecule. If you go one step above, you have the quaternary structure. Basically, when two or more polypeptides join together, such as the case in the hemoglobin molecule, which is a protein in the blood that binds oxygen.
Most proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure.
So now we will talk about the primary structure of a protein. And basically, the primary structure is nothing more than the unique sequence of amino acids. Let's take the lysozyme as an example. This is an enzyme that attacks bacteria, and it basically consists of a polypeptide chain made up of 129 amino acid. Now, the precise primary structure of a protein is determined by the inherited genetic information. Basically, the DNA tells the cells what is the unique sequence of amino acids that each protein should have. And that is the primary structure. Now, this primary structure is very important because even very slight changes in it can alter and have a negative effect on the conformation of the protein. And if it alters the conformation or the shape of the protein, this will also affect the function of the protein. An example is in individuals that have sickle cell anemia or sickle cell disease. Basically, they have an abnormal hemoglobin, which is an oxygen-carrying protein, and this develops from a single amino acid substitution. So only one amino acid will cause this sickle cell disease. Now, the secondary structure. Basically, a secondary structure of a protein results from the hydrogen bonds that occur at regular interval along the polypeptide backbone. We have two kinds of these structures. We have the alpha helix, as you can see from the figure, and then we have the beta pleated sheets. And these are the two known secondary structures of a polypeptide or a protein.
In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself, or where two regions of the chain lie parallel to one another.
Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone exert a strong pull on electrons, giving them partial negative charges, and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself.
Next comes the tertiary structure, and basically this results in interactions between the R group and R group of two neighboring amino acids or two distant amino acids along the polypeptide chain, as well as interactions between R groups and the protein backbone. Now, there are different kinds of interactions that happen to cause this tertiary structure. For example, we have hydrogen bonds. We have hydrophobic interactions between the nonpolar amino acids, and we have the van der Waals interactions among the hydrophobic R groups as well. And you can see different kinds of these interactions on the figure. Another important interaction is what we call the disulfide bridge, and this basically occurs between the two SH molecules between the cysteine monomers, and this stabilizes the structure of a protein.
Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups.
Finally, we come to the quaternary structure. Now, the quaternary structure basically results from the aggregation of two or more polypeptides that come and join together.
Some proteins consist of two or more polypeptide chains. The fourth level of protein structure, quaternary structure, results from the combination of two or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of two kinds of polypeptide chains. Two of each, a total of 4 chains, make up each hemoglobin molecule.
The chemical and physical conditions affect the protein's conformation, and as I mentioned, the conformation is vital in determining the protein's function. For example, if you alter the pH, the salt concentration, or the temperature, or other factors, this can alter the protein's conformation. And if you change the protein's conformation, we are denaturing the proteins, because we have disrupted the hydrogen bonds, the ionic bonds, and the disulfide bridges that maintain the protein's shape. Now, there are different molecules, different chaperonin proteins that help the protein fold onto its proper conformation. Now, most of these proteins appear to undergo an intermediate step before reaching their mature configuration. And the folding of these many proteins is protected by what we call the chaperonin proteins that shield out bad influences. You can think of it as basically a changing room. You go inside and then you change your clothes and you come outside. A chaperonin is the same thing. A protein goes inside the molecule, the chaperonin molecule, folds on its proper conformation, and then leaves this chaperonin protein. Now, the chaperonin does not directly play a role in the folding of the protein. It simply provides it with some privacy or a favorable environment in order for it to fold on itself. And on this slide, you can see the types of proteins and some of their functions and some of the examples on the right hand side. And with that, we come to the end of today's lecture. So please stay tuned. In the coming lectures, we are going to discuss how a DNA molecule determines the sequence of amino acids in a protein molecules.